Hello. Ay. Tu. Hello, I'm Mariana. Hello, I'm Mariana. So close, like 3 fingers. Okay. No, I think that is fine. The lights? Yeah. Okay. Do you think? I never use it, so okay, I can try. Oh, hello. It's better. Okay. Okay, thank you. Mm-hmm. Okay.
Good morning, everybody. Thank you for coming. If you are on Zoom, please, you're muted and you need to stay muted. And if you're in the room with a cell phone, let me do something really quick here. Please silence your cell phones, will be greatly appreciated. Henry is going to make an announcement this morning.
Good morning. My name is Henry Logan, and I have the privilege of chairing the ILR board. Thank you so much for joining us for this 7th of a wonderful series. Walter, I want to acknowledge you for putting together an amazing series of student presentations. We had long conversations about those in the hall. They've been that good. And I understand you've promised to do it next summer too. So we are thankful for, that's called pressure. I wanted to also let you know that on September 21st, Sunday at 2 o'clock, we will have our kickoff for our fall courses. We have courses this fall that are very impressive, all the way from AI to economics to African art to short stories. It's going to be a fabulous fall term. At 2 o'clock on September 21st, we're all going to convene in this room, and we're going to hear from some of the facilitators who will describe in more detail these courses that start on September 29th. Am I right, Pam? September 29th. You will be able to register for the courses on site, which is very helpful, I think, for many of you rather than having to mail it in. So please save the date. I have 2 cards left out of 100 that I had printed to give to people to remind about the kickoff. Here, September 21st, 2 o'clock, and then we'll go out into the upper commons and have some wine and cheese. So thank you, and I'll turn it over to Walter.
Good morning, Chocoholics. Glad to see you here, and I don't remember having promised Henry that I would do this again next year, but we will talk. So welcome to the 7th of our 6 speakers this year, and today it's going to be about chocoholics, and that's all of us, I think, and the danger that's posed and how Mariana is going to fix it all. So Mariana is from Colombia, and she's doing her work on chocoholics. Actually, it's on the black pod disease and cacao pods. So welcome, and we're looking forward to how you're going to solve this dangerous problem we all hate.
Can you hear me well? Okay, so first, thank you for the invitation. I'm so excited, and this is my first time giving a talk for more than 12 minutes. So I hope you don't get very bored with me. So I'm PhD candidate, and Dr. Goss is my PI, and I want to share with you like a broad vision about my research. So first, I want to talk about biotic expressing plants. So as you know, we have some challenges, so plants have some challenges leading with viruses, bacteria, fungi, oomycetes, and some other like nematodes and insects. So today I want to focus in this group. This is the fungal and oomycete pathogens that affect plants and cause losses and also cause reduction on yield and also have a big impact in communities. And also one thing that is important to mention is like the 85% of these plant diseases are caused by this group of pathogens, fungal and oomycete. So I want to give you just one idea of how we can see or how is the infection process when we have a true a true fungi that is here on the top. I will show you true fungi, and here on the bottom, I will show you some pictures from omycetes. So here we can see an spore that land in the surface of a plant, and the spore germinate and create this structure that we call appressorium. And this appressorium is a special structure that allows the pathogen to penetrate the cuticle of the plant and can continue with the infection process. And this is very particular, and this structure is shared in this true fungi that is Magnaporte grisea, but also we have the same structure in Phytophthora infestans, where we can see here the spore, and we can the zoospore here. We can see then the formation of the appressorium. So with this appressorium, this will penetrate to the host. And then here I want to show you Bloomeria graminis or barley powder mildew, where we can see the colonization of the pathogen inside the cells. And you can see how the pathogen that is in green color is inside of the cells. And here you can see Phytophthora infestans colonizing this plant. And you can see maybe these tiny fingers. This structure we call a haustorian, and the haustorian is an structure that allows to interchange some proteins or effector release some proteins to the host cells in order to manipulate the machinery of the host cells. And you can see here this finger type, and here you can see some finger types. So this is inside the cell. So I want to talk a little bit more about omycetes. So omycetes also can infect like a broad range of different hosts. We have here some diatoms, and we have here foxtail millet that is infected with Sclerospora that is also a omycete. And we have here, this is a basil leaf, and we can see here the downy mildew that is caused by Peronospora. So you can see these structures here are the body of the sporangiophores of the Phytophthora, of the Peronospora, sorry. And this is the typical growth of Phytophthora. This growth that is chrysanthemoid, I don't know if this is the correct name, chrysanthemoid. And you can think that this is a true fungi, but it looks quite similar. And also we have here some structures, some oospores, and also here we have some conidiophorus. So this is all to show you that it looks quite similar, the behavior of the omycete, the structure, the hyphae, and the reproduction mode is quite similar. But if you can see, they are very distant in the phylogeny. So we have here the true fungi that are close related with animals, but we have here the omycete that belongs to the Straminophilus. So we used to work together, these two big groups, true fungi and omycete, but it's very important to remember that they are very distant in the tree of life, to say that. What are the difference between omycetes and true fungi? So first, omycete, we have zoospores, but we have this heterocont behavior. We have two different structures that allows the spores to move, but we have just one flagella for the opistoconta, for the true fungi. And also, one very important feature is like the cell wall for the true fungi. We have some glucan and some chitin. But on the other side, we have for the omycete cellulose that is quite close to the green algae or brown algae, so more close to the plants. But the true fungi are more close to animals. So just to remember, please, that bats are not bears, dolphins are not fish, and omycetes are not true fungi. So first, I want to talk here about um, potato. Maybe you wonder why I'm talking about omycete. What is the importance of mycete? So to maybe give you a little bit of context, so maybe you are familiar with the Irish potato famine. It was caused by Phytophthora infestans. It was a devastated event, mainly in Ireland. So between 845 and 1800, sorry, 49, we have this devastation. So around 1 million people died, 2 million people migrate to America, and it was the main problem here. It was like potato. It was introduced as a staple crop for European countries, but mainly in Ireland they established some potato variety very homogeneous. They don't have like this diversity of different potatoes. When this disease um, start to propagate, it was devastating because they have the same kind of or the same genetic background, so they don't have diversity, and this will cause more impact in Ireland. So this is the kind of symptoms that Phytophthora can cause, and this is completely inedible. So, and also the population of Ireland suffered dramatically. So from 8 million people there, uh, the decrease it was like almost to 4 million people. So it was devastating. And even nowadays, they are still recovering. Mm. But now I want to switch to my crop that is cacao. So cacao is a very important crop and is a superfood. And cacao is present in 61 tropical countries and is very important for the livelihood of small farmers around the world. And the production of cacao beans could reach 5 million tons per year. And the main producers of cacao beans are West Africa countries with almost one third of the production, and 22 is produced by Latin America and 6% from Asian countries. So also cacao is very important because it's full of antioxidants, and we can track like more than 100 use as a medicinal purposes, and also of course as delicatessen, as a gastronomic, a dessert, that everything that you want to put uh, in the sweets, dessert, or any kind of food that you want. And also it's very important for cosmetic, as you know, maybe some product for that we like to use, shea butter and this stuff. Um, but cacao is also the main challenge that we suffer, one of the main challenges, because cacao is facing too many challenges nowadays because of the climate change, because of the aging of the crop, but also because of this disease that is black pod rot. And this disease affects the fruit, and it start with some dark lesions, and by the time this dark lesion can cover completely the pot, and when it's completely covered, could reach a mummified state, and we cannot harvest this. So this disease is caused by different Phytophthora species. So we have different species that can associated to this disease. We have Phytophthora palmivora, we have Phytophthora megakarya, Theobromycola, and others. So just I want to highlight the most prevalent, Phytophthora palmivora and Phytophthora megakarya. So this is a typical disease of, well, this is was inoculated in the lab, and this is how it looks, the disease in the lab. We have here Phytophthora palmivora. Phytophthora palmivora is a broad host range, so Phytophthora can cause diseases in papaya, coconut, oil palm, durian. and many other crops, around more than 200 crops, more than 200 hosts. And this is quite large distributed in the world. Um, on the other hand, we have Phytophthora megakarya. Megakarya is uh, a pathogen with a that is just cacao host infected. So, and also is confined. Megakarya is just in West Africa. So you can see here the hair is just here in this area. So it's a narrow host, and also um, the difference between these two is like nowadays Palmyra is less aggressive if we compare with Megakarya. The loss that we can have with Palmyra could be 30%, but the losses with Megakarya could reach 80%. So it's very aggressive, and also it's important to mention like the size of the genome of this pathogen, Phytophthora megakarya, is almost the double of the size of Phytophthora palmivora, and also some effectors or some proteins related in virulence is almost the double in megakarya compared with palmivora. So for the first part of my project, I want to talk about the differential response of different cacao clones against different Phytophthora species. So what we did, we used different cacao clones. We used the germplasm collection in Catie in Costa Rica. So we use these 20 different cacao clones, and we use inoculation technique in the lab. So we use different Phytophthora species. We use 7 Phytophthora palmivora isolates. We use 2 Phytophthora megakarya, and we use just 1 Phytophthora theobromicola. This species, Phytophthora theobromicola, is just located in South America. We have reports in Brazil and also in Colombia. So with these isolates, we perform the inoculation to each one of these clones. And here is important, I don't know, you can see here we have 2 reference. Pound 7 is our reference, is a cacao clone resistant to Phytophthora palmivora. And UF273 is our susceptible reference. This will be important later. So one thing that is very important is like we select these cacao clones and we select this reference. Consider the resistance or the performance against Phytophthora palmivora. So this is how we evaluate the response of cacao to Phytophthora. So we have a collection of the collection of isolates, and this came from different infect pods or isolates in collection storage, or we have in the lab. So we grow these 5 days in dark, 5 days in light, and then we put water, uh, cold water, and we perform the incubation, and we release the zoospores, and then we measure the zoospore, and we have this standard zoospore suspension for each one of our inoculation. So we inoculate in 6 different points in each one of our cacao pods. We use 6 points, and we incubate these during 4 days. And after 4 days, at 25 Celsius degrees in dark condition, we measure the lesion. And this is the way that we perform this evaluation to determine the lesion side of each one of these cacao clones when we challenge to each one of with each one of the isolate that I mentioned before. So this is the kind of symptoms that we obtain in the lab. We use this 1 centimeter as a reference. So we use this paper to put the drop, the spore suspension solution, to allow to stay in the pot. If not, it will fall down. So we use as a retention system this square. So after 4 days, this is the kind of lesion that we can see. So this is a very susceptible cacao pod. maybe is one of these UF 273. But on the other hand, we have some cacaos clones that perform very well. They have very small lesions after 4 days, or even we can see here these tiny dots that we can see. Maybe we have here some defense or some defense response. So we have a very contrasting behavior here. So when we check the differential clone response, we have here our different cacao clones, and we have the lesion site of each one of these. So we can see like we have here a reference, a resistant reference, pound 7, and we have a susceptible reference that is UF 273. So you can see here that even we have some cacao clones with bigger lesion sites than the reference, but on the other hand, we have here some cacao clones that could have even a smaller lesion compared with the resistant one. So that means that here we have a good candidates that perform better even than our reference. So when we compare between two different Phytophthora isolates, this is a reference. GH49 is the reference palmivora isolate. We can see a median of 14 millimeters, but also we have another Phytophthora palmivora isolate that is from Colombia, and we have the median side of 35. It's quite is more aggressive if we compare with the reference. So that means that we have a different behavior even when we use the same species. We use both as palmivoras, but we have a very different and interesting behavior here. So when we compare now the clone defect by clone, so we can see here this is organized, but which one is less susceptible and which one is more susceptible. So we have here is the same, quite of the same that I explained before. And when we compare the isolates and we want to see how they are performing, so we have here GH49, that is our Phytophthora palmivora reference, and we have here our Phytophthora megakarya reference. So this isolate is the most aggressive one, and it was something that we expect, that we can see like we don't have any clone that could have resistant to megakarya, but we have some clones that could be less susceptible to palmivora. So this is quite messy, but I try to explain. So we have in orange this line that belongs to megakarya. This is one isolate from megakarya, and this is the other megakarya isolate. And all the colors in blue belongs to Phytophthora palmivora isolates, and the green one is the Theobromicula that is from Brazil. So you can see here that megakarya is the most aggressive one when we compare against all the different clones that we have. It's almost always more aggressive than when compared with the other isolates. And if we compare between two megakarya, we have an interesting pattern. We have here a more aggressive isolate, but we have here a less aggressive isolate, even when we have the same species. So this is very important to understand that we need to be careful and we need to identify and we need to track the behavior of the isolates in the field. We cannot use just one reference. If I use this reference, this megakarya, to perform all my analysis, I will lose a lot of information because in the field or in other areas, we can have more aggressive isolates. So this is very important when you are doing the screening to obtain resistance, that use more than one isolates, and you need to use a variety of in different areas, isolates that came from different areas or different countries. Yeah, and when we compare with Phytophthora palmivora, this is like the 7 isolates of Phytophthora palmivora, we can see some interaction. So we can see like Phytophthora palmivora, this isolate, that is the Colombian isolate, is very aggressive when we inoculate to RF31. This is my cacao clone. So there is some kind of interaction here, and it's quite interesting that this isolate it was obtained from Colombia, and also RF31 is also a cacao clone that is established in Colombia. So maybe this cacao clone is not a good idea. It's not good idea to have this reference, this cacao clone in Colombia, where we know that is very susceptible to an isolate that is there in the country. So this is another important thing that we need to consider. Like we need to evaluate and use the isolates from the country if we want to put their new cacao clones. So we need to test with different isolates, but mainly from the isolates that are in the same area. So we can see here again that we have some clone and isolate interaction where in some particular clones, Phytophthora palmivora could be more aggressive than even a reference that is Megacarya that is the most, in general, the most aggressive one. So if this is the broad vision of where we have cacao. So all the colors belongs to the production of cacao. So as I mentioned, we have a tropical tropical crop, and we can see that all this blue area is that where cacao Phytophthora palmivora is present. We have here one isolate. One isolate. This is from Colombia, and this is another palmivora from Ghana. This is another from Papua New Guinea, and this is from Indonesia. For Phytophthora megacarya, we have one from Ghana and one from Nigeria, and Phytophthora teuromicola is located in Brazil. So it's very important to continue doing this type of research to evaluate different isolate from different places around the world, and also to continue integrate on to have a broader Phytophthora associated species. Yeah, so genetic diversity is very important and have a broad impact in the breeding process. And also we have some important components, the host and the pathogen, significant influence the interaction or the disease progression in cacao. And also the virulence of this black pod disease is not just associated with one specific species or one specific isolate. And yeah, then I will show you the next part of our project is the characterization of these two species, Phytophthora palmivora and Phytophthora megacarya, and we check the gene variation using a transcriptomic approach. So here, I want to show you, this is the general process of how the pathogen could colonize. So we have the zoospore, and then we have the appressorium here, and the appressoriums go inside the cell plant, and here we start to release some proteins to manipulate the host cell, and if the plant can recognize the pathogen or the protein of the pathogen, we have a cell death because we have this recognition. But if not, if the plant doesn't have the mechanisms to recognize this protein, so we have a proliferation of our pathogen, and the pathogen can go between the cells or inside the cells. And here they use the Austorian, that is this finger-like structure that can colonize inside the cells. And at the end, the pathogen will develop again this sporangiophora, where they can release again the zoospore and continue the cycle. So just to reminder that we have these two fogs. We have palmivora that is broad host, and we have our megacarya isolate that is just a cacao host. So we perform the transcriptomic analysis. So I don't know if you are familiar with transcriptomics, but what we did, it was like we isolate the RNA from the sample, we send to sequence where they just fragment the RNA in small pieces, they convert to cDNA, and then use some adapters to perform and to do the sequencing process. So at the end, what we want to see with this approach is we want to identify the genes that are important in this interaction, and we want to see genes that are highly expressed or or have lower expression. So we want to obtain this kind of figure to try to analyze and identify important genes in this infection process. So what we did. So we use now 10 different cacao clones and consider the first part of our project. We identify a uh, differential disease progression of these cacao clones. So we have a very susceptible one, a very less susceptible, and we have intermediate cacao clones. So we tested against two different Phytophthora species, one Palmyra and the other one is Megacarya, and we use water as our control. We used the experimental design. It was 10 cacao clones, 3 treatments, that is these 2 isolates and the water, 2 time points after 12 hours and after 48 hours, and we used 3 biological repetitions. So what we did, again, we use a zoospore suspension to inoculate the cacao pods. So we inoculate the pods. Oh. So we inoculate the cacao pods, and after 12 hours, we collect samples. And after 48 hours, also, we collect samples. With these samples, we freeze dry during 1 day, and then we perform the extraction of RNA, and then we send to sequence. And then with this information, we perform a analysis where we clean these reads, we did all these pipeline, and at the end, what we did, it was to obtain the differential express gene, which gene are turning on and which gene are turning off in this interaction. And at the end, we want to see which genes are particularly upregulated is our main goal. So this is the result where we can see the variance of these samples. So here we have GH49, the mycelia. This is just the information from the pathogen, our reference, that is Palmyra here. And we can see here the information from the other clones. And this is a variation. So this is the most susceptible one clone, and this is the most resistant one. So you can see like the color, it has this variation against this variation where IMC is here, here, UF is here, here. So we can see this different range variation here in our samples. But what we have for Megacarya is what we have 2 different groups. One is for our mycelia, that is GH34, that is a completely different behavior that we have with all the other cacao clones after we collect the samples after the 48 hours. Sorry, here I will show you just 48 hours. And we can see this contrasting behavior. So we don't have this variation that we saw in our Phytophthora palmivra. And after our analysis, we identify particular genes that are upregulated. So one thing that is I forgot to mention is because after 12 hours, the infection process is so low, or the amount of information that we can recover from the pathogen side is so low. It's almost between 0.03% or 0.02% of the all information that we recover belongs to the pathogen. So that means that it's very hard to do analysis at 12 hours. So we use just 48 hours where we obtain more information to perform the analysis. So what we have here upregulated genes that are important just when the pathogen, this Phytophthora palmyra, infect IMC60. So these are very important genes that are exclusively associated in this interaction. But we have other genes. Here we have three genes that are important in the interaction between Phytophthora palmyra and all of them. We can talk about core genes or um, core genes important in this infection process. But also we have another set as these other three genes that are important to infect IMC60 and to infect L-cysteine. But what we can see with Phytophthora megakarya, the behavior is very different. We have more upregulated genes, and this is because we recover more Phytophthora megakarya reads, because as I mentioned, it's more aggressive. So this pathogen could colonize faster than Phytophthora palmyra. So then we recover more information. We have more reads of this pathogen. But it doesn't mean like Phytophthora palmyra don't use other genes. Maybe the pathogen use more genes, but because of the amount of information that we recover after sequence, we cannot extract all the things that are involved there in this interaction. So here we can see we have 131 genes that are important in the infection process when we challenge all these different cacao clones against Phytophthora megakarya. So the amount of upregulated genes is huge. And now also we can see some particular genes. For example, here we have 4 genes that are very important when the pathogens colonize SK6 and CATIE. So this is like I didn't mention, but this is like a Venn diagram. But this is a different way to see a Venn diagram. If I go back, this is unique, and this is all the center of the Venn diagram with all of them are, with all of the different cacao clones are confined in the center. So this is a different visualization of a Venn diagram, basically. And so continue with the last part of my project is to characterize these two different pathogens using a capture sequencing or a target sequencing. So first, before continue, it's very important to talk a little bit more about plant immunity. So here you can see a landscape, a beautiful landscape. This is from Colombia, where you can see some coffee, we have some plantains, we have some cucurbits here. We have this beauty, and you can see a healthy, or this is landscape, what you can see here. This is a very healthy, right? So by default, the plants or the state is healthiest state where we can see in plants. So disease is something that is abnormal in plants. The majority of time we can see just healthy plants. But what we cannot see is this. This is a battlefield. This is what is happening right now in the field, in our crops, where we can see some pathogens try to colonize the plant, that is this castle, that they want to go inside because they have some resources, they have some nutrients, some have all these things that the pathogen desire to continue propagating. So this is what we cannot see in the field, but it's happening all the time. All the time, plants are fighting against pathogens. So I want to talk a little bit about this plant immunity, how it works. So the pathogen reach the surface of the host cell of the plant cell, and the pathogens can be recognized by the plant. We have some receptors here in the surface of the plant, and this receptor will recognize important proteins or important elements from the pathogen. When we have a recognition, we have a defense response that reach to this resistance. But by the time of the evolution, the pathogens start to deliver some proteins inside the plant cell. And these proteins or these effectors will cut this recognition. This, this will cut or will avoid this recognition. And when the plant cannot recognize something that is external to the plant itself, so we have susceptibility. This is the most important concept in plant immunity, the recognition. If the plant can recognize the pathogen, we have resistance. If the plant cannot recognize, we have susceptibility, we have disease. But also, the pathogens evolve and create new ways. The plant evolve and recognize these protein, these effectors. So when the plant recognize using these R proteins that target specifically these effectors, we have again recognition. And with the recognition, we activate this defense response and we have resistance. But at the same time, pathogens evolve, and okay, then I will release a different set of effector or different set of proteins associated to virulence to cut again this signal. And when the pathogen could block this defense, we have again susceptibility. And again, the plants develop new R proteins that can recognize this new set of effectors. So this is a beautiful dance in the evolution between plants and pathogens. And this is something that is happening all the time. So here just to highlight the importance of these proteins that use the pathogen to manipulate this mechanism, the whole cell mechanism or machinery. So this could be quite overwhelmed, but this is just I want to show you what could happen inside the plant cell. This is everything that we can see. Here is all the different process of the pathogen, the zoospore, then reach the surface and form the austoria, the appresoria, the appresoria can continue with the hyphae, and then the hyphae develop this structure, this is austoria, where they can release the proteins of the effectors to manipulate the host cells, and again, so so the pathogen is fighting so hard also to try to reach or to obtain the nutrients from the host cell. So we have different protein related, virulent related proteins. We have some proteolytic, we have some lipid membrane modifiers, we have some transporter, we have some proteins involved in the redox of the host cells, and also we have some proteins involved in signaling, and also some involved in osmotic stress. So all of them are different pathogens proteins that help to manipulate or to reach the goal of the pathogen that is colonized and continue the cycle of propagation through the release of zoospores and so on, so on. So here we want to characterize the population. We have a collection in our lab. So what we use our collection. We have 87 isolates from this guy, that is Phytophthora palmivera. This is the representation of where we obtain each one of these samples. So we have samples from US, we have samples from Colombia, Ecuador, Trinidad, Ghana, Malaysia, Japan, and Papua New Guinea. But also here I want to mention, like, we have mainly cacao isolates, but we have some from other hosts, just as a reference. And for our megacari isolate, we have 82 isolates. And as I mentioned, it's just located in West Africa. So here is important that you can remember that we have isolates from Ghana that is in the here in the left part. We have Nigeria that is in the middle of these two other countries, and we have Cameroon that is in the right side. So what we did, so we obtained the mycelia of each one of our isolates. And also, on the other hand, we have these different virulence proteins. So we develop probes to target each one of these genes. So we have 2,198 genes related with this process of virulence that I mentioned in the previous slide. So we have this probe set, we have the mycelia, and what we want to do, or our goal here was to target just these specific genes. So here we don't use a whole genome sequence. We use just sequencing, sequencing just for each one of these genes. So this is what we call capture-seq or target sequencing, because our goal is just to analyze these specific protein virulence associated. So we perform the bioinformatic pipeline, and we clean and we map, and we just obtained the SNPs, that is the single nucleotide polymorphins. We recover this to run our population analysis. So what we obtain here now is we have our cacao isolates. We have 66 cacao isolates that all of them are in here, in this area. On the other side, we have this durian isolate and one cacao isolate that is here. So we can see a clearly differentiation between these two groups, between this cacao isolate and between this durian and cacao isolate. But on the other hand, we have in the middle this group, 5 cacao isolate, and this non-cacao, non-durian isolate. When we analyze the ancestry of these all these collection for Phytophthora palmivora, we can see a big group that belongs just to cacao, and also we have the group that belongs to durian, and this is non-durian other host, non-durian, non-cacao, and this is quite correlated with the PCA that I showed you before. So that means that we have here three clustering of our population, of our Phytophthora population. And then, on the other hand, we have our isolates from Megacarya, where we can see here one group that belongs to Ghana. Do you remember that Ghana is in our left side? We have here our group that is from Cameroon, this green one, and the right side. So we have two very contrasting behavior. Here is Ghana, here is Nigeria, here is Cameroon, and in between we have the Nigerian, but we have one group of Nigerian isolate that cluster with Ghana isolate, and we have some Nigerian isolate that cluster with the Cameroon. So if you remember, Nigeria is the country that is in between these other two countries. So that makes sense that some of the isolates that are maybe collected and more close to Ghana are clustering with the Ghana isolate, and the other ones are are closest to the other Cameroon. And when we check the ancestry, we can see two clusters. So we have Cameroon, that all of them are yellow. We have the Ghana isolate that all of them are here in the purple color. And we have the Nigeria, where we have the combination of these two different clusters. So now, uh, to finish, we want to integrate these two approaches. So we have the genes that are important in the infection process, the genes that are upregulated, but on the other hand, we have the information of each one of our isolates, and we have all the variants of each one of these important, each one of the genes of each one of the isolates. So now our goal, it was to combine these two different approaches to identify if I have the list of my genes that are important in the infection process, I want to check what happened here in our information, in our genome information, to identify the candidate genes that are important in the infection process for cacao, which genes we have that are more relevant or are key in this infection process. So we we identify some genes, we check the SNP associated or the SNP variation, and here we have 8 different virulence protein related. We have secreted proteins, RLXR. We have glycoside hydrolase family, and we have elicitin, glycine, and some transglutaminase elicitor. So here we can see like the main variation in our samples correspond to this area where we have isolates from Papua New Guinea, Malaysia, and Indonesia. And it's quite interesting that the same isolates that are here are the isolates that are from cacao. If I can go back a little bit here, so those are the isolates that are from cacao, but don't is sorry, they are not associated with the main group. So here, these 5 cacao isolates are the ones that correspond to the Malaysia, Indonesia, and Papua New Guinea. And this is quite interesting for us because those are the isolates that have some variation. So this will be very important for us to identify and to see if this SNP variation is associated with a real allelic variant or we have some copy number variation. So this is the ongoing project. So we want to later identify if we have this allelic variant and to try to determine which genes are important and how these genes are different in our collection. And with that, uh, I want to mention like this is an ongoing project, so um, we want to continue doing this validation of these key effectors that we just identified. We want to expand also the sampling of our Phytophthora collection to have more hosts and more regions. But because as I mentioned, the diversity of the pathogen is very important and it will be relevant to continue with the screening of the breeding process. And also we want to explore more this cacao gene and how this cacao gene responds to the infection. And with that, I want to acknowledge my PI, Dr. Erica Gauss, my committee members, and also our postdoc Ina, Josh, and our lab members that support this research, and the funding agency, Mars, that funding the cacao shipment and also the sequencing, and Katia, because they sent all these cacao pods where we use in our lab to do the inoculation, and all our other collaborators that submit the isolates to have in our collection. And with that, I want to say thank you. If you have questions, I'm more than happy to answer.
Do we have questions?
I'm not a pathologist, so I'll leave it to some of the other people. But was the severity of the disease at all related to where you saw more variability? So, no, the question is really, is you got some variation in these different countries, I gathered from your, and did you have any inkling that if there was more variation, that there was less severity of the disease, in other words, it was wiping out, or is that totally unrelated? And do you have any indication about the severity of the disease in those regions of the world?
Well, we know that in Africa the severity of the disease is higher than any other place in the world because we have there megacarya that is the most aggressive species.
So it's really the more aggressiveness.
But it doesn't mean that we cannot have some Phytophthora palmyra that also could be aggressive because as I show you, we have one Colombian isolate that could be even more aggressive than megacarya. So it's not like, in general, Phytophthora megacarya is more aggressive, but also we have some particular isolate that could reach this level of virulence, of aggressiveness.
So is the chocoholic group of us going to be in real trouble because this stuff is going to go around the world, and or are you going to fix it somehow, and if so, how?
I don't know if we will fix it because it's quite hard. We try to identify and try to prevent. So this project is part of another big project that is the preventing breeding. And I don't show, but what we are doing is to develop this model. It, is develop this model because until now we don't have any material that could be resistant against Phytophthora megacarya. But my megacarya is right now just in Africa. But at some point, we know that megacarya will reach America, right? And we don't have any resistant material. So we are doing this screening process to evaluate which cacao clones could be less susceptible to megacarya in order to, anticipate this problem. But unfortunately, we don't have right now any material, but we have some good candidates, and the breeders can work on those good candidates and do the breeding process to have a resistant material at future.
So you're looking for the different kinds of the cacao plant that would be less susceptible to the diseases? Is that where we're heading?
Yes, exactly.
And what fraction of the 6 million tons or whatever it is of chocolate that comes around, what fraction of it is being lost due to the pod disease? And are they doing anything? What, you go out in the field and you see a black pod and you just don't pick that one, or what do we do?
So the thing is that the losses could reach 80% of your farm. You can lose 80% of the entire production. So unfortunately, if we don't have like resistant, we need to just take care of the farm, like remove all the infected material. You cannot leave it in the soil. You need to pick it and remove it, and you need to prune in the trees, and you need to be sure like you don't have all these uh, damp soil. You need to have like dry soil, or you need to rise the bed of the trees, to try to avoid this water because Phytophthora loves water. So this is the kind of things that people can do, and also don't move material between regions because when people love to move material, oh, this is great in my farm, is performing really nice, but you don't know if when you move, no, you don't know for sure. When you move your plant, you also move the pathogen associated to this plant. And maybe when you change the environment, maybe in your farm it's not a problem because it's a very dry or something like this. But if you move this material to other farm, it's more wet, or we have more shading. Maybe the pathogen could reach the perfect condition to propagate. So this is what happened when people move material without caution.
Okay, any other questions? Excuse me.
I might have missed this, but um, how are the plants normally fertilized? I don't know if cocoa plants are self-fertilized or if they're male and female. How are they normally fertilized, and are the environmental conditions different in different places so that it would be more susceptible in with some environmental conditions than others?
Well, for the first question, I don't know, to be honest, because I'm not work directly in the field, so I'm not aware about what is the fertilization that people use in the farm. But for the second question, yes, absolutely, because the condition, the weather, the climate, it will define if the pathogen could have the optimal condition to grow. So yes, and because of that, I mentioned like you want to avoid the water for Phytophthora, and you want to remove all the. Also, sometimes people just cut a lot of the leaves or the branches in order to have more exposed crop, to have more light. Because if you have very dark and very wet, it will uh, reach, it will increase the humidity, and this is a perfect environment for Phytophthora. So this is the kind of thing that people are doing, like try to keep it dry, your crop. But yeah, definitely condition are
So
determinant.
let me ask it this way. Um, the flower blooms before you make a pod. Are the flowers fertilized by bats or insects or birds? Yeah. Yeah. How are the flowers fertilized? And can that contribute to the movement of the pathogens from plant to plant?
Yes, it could happen that also the pollinizer could play a role in this dissemination of the disease of the pathogen. Yes, could.
Yeah. But but I don't understand how the flowers are fertilized. Are they self-fertilized and you don't ah so that would be a question for somebody to look at. We see all of these hummingbird things going around and they have special beaks for special flowers. So anyway, something to consider. Any other questions that we have? Any questions on Zoom? No questions on Zoom. Thank you very much for coming, and I hope that we can fix the cacao problem.
Thank you.